Hello students, welcome to EPG Pathshala. I am Dr. Amarjit Singh from Department of Physics, Himachal Pradesh University. Today we are going to discuss about the module Surface Physics and Characterization Techniques from the paper Solid State Physics. So students, let us see what we are going to learn in this module. So we are going to learn about characterization of surfaces, interfaces and thin films. So in this we will pick up the two of the most popular techniques, and one is that we will learn the application and the working principles of atomic force microscopy.
Let me introduce you
advantages and
some of the idea
disadvantages
of
atomic
surface
force microscopy
characterization.
So
as we have
scanning
seen that
tunneling microscopy,
learned that
its principles and
surface
applications
of
any substance is not truly smooth, but it always has got some roughness, contaminations and defects. So we need to thoroughly study the surfaces so to understand the science behind nanotechnology and their applications. So first of all, we have to study the surface morphology, how they look like. So some of the techniques which are used is one of the technique is atomic force microscopy, and the other technique which is quite known in this area is scanning tunneling microscopy and scanning electron microscopy, which works at a slightly low resolution. And these are the direct technique where we use a probe. These techniques use a probe method kind of thing, and you can look at the surface in they give the direct view of the surface. But there are techniques which are also which are indirect techniques, such as grazing incidence, X-ray diffraction, X-ray reflectivity, neutron reflectivity. We know that using this probe technique we can probe the surface morphology, but we cannot see below the surface, or if there are several layers and we cannot see the interfaces. So for that, this indirect techniques like X-ray grazing incidence, X-ray diffraction, neutron reflectivity, X-ray reflectivity, they give us very important information of the some interfaces below the top surfaces. So these are just the techniques of studying the morphology and thickness, but there are whole lot of techniques which talks about this chemical analysis, transport and magnetism. We will not touch upon that. So in this module we have just picked up two techniques out of them, and one is AFM and another is STM. So we will talk about in brief about these two techniques. So when idea of a surface comes to our mind, we usually think that surface is a plane. As we see plane of a floor or plane of a water, so this gives us an idea that this is a smooth, a plane surface. But when we see it microscopically, we enlarge our view, that we see that all kinds of surfaces have roughnesses, and none of them is entirely smooth. So as you can see in this diagram, there are pits and valleys and projections. Even a water surface of a still water, it has its roughness, which we call capillary roughness, because due to the disturbance in the medium there are some oscillations on the surface. And also in case of plain solid surface, even if we have a monolayer deposited there, so there is an atomic level roughness. So it depends at what length scale we get that roughness, but that roughness is already there. So for this reason we have a certain morphology associated with the surface, and that surface morphology is extremely important in determining its entire properties. Optical properties, because the reflection depends on how smooth the surface is, and also frictional properties, this tribological properties, they very much depends on this surface smoothness or roughness. So we have to have equipment which can measure this roughness at some nanometer scale or even at the atomic scale if we really want to understand the nano devices or nanoscale phenomena occurring at surfaces and interfaces. AFM and its application. Atomic force microscopy is a high resolution type of scanning probe microscopy that allows us to see and measure surface structure in length scale 10 nanometer to 100 micrometer with high resolution, and it has a lateral resolution of about 30 nanometer and vertical resolution is very good, which is 0.1 nanometer, so even 1 angstrom changes we can see. Unlike an traditional imaging microscope, AFM provides height information of the sample. Almost any sample can be imagined, be it very hard ceramic kind of material or soft human cells or DNA or different biological molecules. AFM is the most common of the scanning probe microscopy and is used in all fields of science as chemistry, biology, physics, material science, nanotechnology, astronomy, medicine, and many more. At these slides, we have, with the help of diagram, we are trying to show that how an AFM works, what is the basic requirement of an atomic force microscopy. So, atomic force microscopy is a probe based technique where we have a very fine probe or a very sharp tip, as you can see, mounted on a cantilever. The tip is so sharp that when you come at the tip of this needle kind of a structure, only few atoms are there which are in close association of this surface. And now on the right hand side here you see that this diagram shows the tip to surface force. So when it comes in the proximity of the surface, this plots that how the force between tip and surface is working. So when it is a long distance, you can see it is almost no force and it comes in the attractive zone. So this attractive which is below 0, so this is due to this van der Waals interactions, which is usually attractive. And when if you bring this tip further near, you will see that this 4 turns into a repulsive kind of interaction. So we shall discuss that how close we can bring this tip to the surface so that we don't break this tip. At the same time, we can measure the surface morphology by scanning it over the surface. On this slide, we show how this AFM work. This principal diagram of this AFM is shown on the right hand side, that you can see that there is a sample holder where you can place the sample, and then there is a cantilever on which this tip is mounted. On top of cantilever there is a laser beam, and this laser beam is reflected from this cantilever top, and this photo detector is there to record the deflection. And there is a piezo control here. I just describe it step by step. The cantilever is bendable structure used to hold the tip. It is basically a spring with stiffness K. The piezoelectric materials are used for controlling the motion of probe as it is scanned across the sample surface. A laser beam is reflected by the backside of the reflective cantilever onto photodetector. The position of the beam in the sensor measures the deflection of the cantilever and in turn the force between the tip and the sample. The feedback loop includes all of the structural elements that are required to hold the probe at the fixed distance from the sample. So here there is a feedback mechanism. When this tip comes to a sufficiently close distance at the surfaces, this cantilever senses these forces. So it depends how close you want to bring it. Either you want to let it in the attractive force zone or a repulsive force zone. So that is given to that feedback is given to the piezo, so that we do not approach this needle any further, because if we press it any further it will break. So this piezo control, this feedback circuit, keeps this position of this tip and the surface constant. Modes of operation. AFM works in 3 modes of operations: contact mode, tapping mode, and non-contact mode. We can see that on the right hand side there is a diagram, and this is force diagram, that when we bring this tip close to the surface, initially it enters into a range of attractive force, and in this attractive force regime, this is what is called non-contact mode. In this mode, the tip is quite far from the substrate, and this van der Waals forces, it is in the range of van der Waals forces. So when you bring it further closer and closer, and then you finally enter into a contact mode, or there the tip begins to, the atom of the tip begins to repel the surface atom. So there the tip is quite close to the surface, and usually we can break the tip if we are not careful enough. In between these two non-contact and contact mode, there is a third mode which is called intermediate, and this is a tapping mode. So in this tapping mode, cantilever, a probe is a tapping on the surface with some given frequency, and the probe to surface separation can be lying from 0.5 to 2 nanometer. So here we show some of the real images obtained by AFM of a surface. Now you can see this, that this is some sort of bacteria or some structure lying on the solid surface on the left hand side. This is a 2-dimensional structure, and height coded in form of color. So you can see that this light color is a larger height, and this dark color is a smaller height. Now with the help of software, this is converted into a 3-dimensional image on the right hand side, which you can see that we can really have a 3-dimensional image of the object. And below it is on the left hand side, this is an image of some indented surface, and you can see that we can really indent the surface at a nano level, and its structure is shown on the left hand side, and on the right hand side we see the surface of one of the bacteria. So basically we can do this AFM imaging on variety of samples, from hard condensed matter to biological systems, and see the surface morphology and their 3D view. Advantages of AFM. So it requires minimal sample preparation, just an available surface is ready for observation, and it does not require any kind of conductive sample or something like this, because you just need to look at the surface where the force between tip and sample measures the morphology. And it provides a 3-dimensional surface profile with the help of software, and XY, the ability to magnify X, Y, and Z axis. It works perfectly well in ambient air or even liquid environment. It is possible to study biological molecules and even living organism. It does not require very expensive equipment. The scanning tip and piezo arrangement is the main component of this equipment, which is not very costly, and due to its small size, it can also be combined with other microscopes or instruments. So it can also work in combination with other equipment, different modes. Okay, so as we discussed about AFM as what it can do, but this technique has some disadvantages also. So it is not practical to make measurements on an area greater than 100 microns. So one has to have a small selected area where one can work, and it has a limited scanning speed, so it is not very fast enough. It takes time to scan a very small area. Images can be affected by nonlinearity, hysteresis, and creep of the piezoelectric material. So here we are using piezoelectric material to scan all this. So the noises from outside can be introduced, and one needs to be careful. And definitely there is a possibility of artifacts in the images due to these reasons. And an AFM image does not reflect the true sample topology, but rather resembles the interaction of the probe with the sample surface, and we use a software to construct the image from this information there. So now let me introduce you with the scanning tunneling microscopy. We have just learned about this atomic force microscopy. So this is another scanning microscopy, but here we are using the tunneling phenomena. It uses quantum tunneling phenomena for the mapping of the surfaces. So tunneling current from tip to the sample is used for mapping. Current is exponentially dependent on the distance between tip and the sample, and raster scanning gives a 2D image in this one, and feedback is normally based on constant current, thus measuring the height of the surface. So it is slightly different from AFM because here we are using the tunneling current, we are using the quantum phenomena, and we need metallic samples and metallic tip or semiconducting samples so that the current transport is possible. Okay, so what are the advantages of AFM? So it gives high resolution scanning, and atomic and even subatomic level imaging is possible. Atomic scale represents the ultimate resolution in terms of chemical compositions of the reactions. How we are able to probe the atomic scale? The single atom scanning, due to the tunneling mechanism, which is sensitive to the tip sample distance, is the key element here. Only the outermost atom on the tip is involved in the movement. So basically, as the tunneling current is very sensitive, it is exponentially dependent on the distance between sample and the tip. So actually the tunneling current decreases very sharply if the tip moves away. So it is extremely sensitive due to that reason, and it is feasible under UHV condition, ultra high vacuum condition, and also we can work in the ambient conditions. And it can give the direct imaging of the atomic structure on a plane, unlike AFM where the in-plane resolution was weak. It has a very strong in-plane resolution, and in situ characterization of surface reaction on atomic scale is possible by potentiostat for biasing the sample, and feedback loop data collecting device computer. So you can see that this arrangement is almost like AFM, except the fact that here we are not using any laser or deflected spot, and also the tip which is required in this kind of arrangement has to be a metal tip, because here we are measuring the current between the sample and the tip, and this current is a tunneling current. So this tip is holded slightly above the surface, and the sample below should also be conducting, either it is a semiconducting sample or it is a metallic sample. Then we can do this STM. So on non-conducting samples we can't do it. So this tunneling current is measured as we do the scanning on the surface. So this tip is scanned in a special fashion, what we call raster scan, that it goes from one position along a line and then it slightly comes down and comes back just below that line. So then in this way we can scan the entire surface and map the value of current. Now here we have to understand, as this current is a tunneling current, so the current depend on the exponential of the distance between tip and the metal. So it is very sensitive to the distance between metal, tip and the sample. On this slide we see this diagrams. Here we on the left hand side, in the middle, and this right hand side C. The C diagram is the actual diagram of a surface taken from an STM device. Now here you can see that we can really probe the position of atoms on the surface. So this is in contrast to AFM, where AFM has a very low lateral resolution. Here we can really see the positions of atoms. This slide also shows this 7x7 reconstruction, which is quite famous. We were talking earlier that there is a reconstruction mechanism at the surface. So this is called 7x7 reconstruction. As you can see in the middle, that this unit cell now is made of 7 atoms along one axis and 7 atoms along another axis. So this is surface reconstruction we can directly see using the STM on a silicon surface. STM does not probe the nuclear position directly, but rather it is a probe of the electron density. So STM images do not always show the position of the atoms. As we can see in this diagram, the graphite layer comprises of totally conjugated carbons as demonstrated. The STM images the conjugated pi electrons which are delocalized among the plane. A hexagonal unit cell of graphite has 2 atoms in its basis, but STM only shows one of the 2, forming a triangular lattice as shown in the figure right. Imaging of angular momentum. So here this is another interesting study of STM, that we can really image the angular momentum of orbitals in an atom. Experimental images of a silicon surface imaged in dynamic STM mode. Different radii of atomic images in A to C are explained by different higher angular momentum tunneling states. Images taken with tips made from different elements, A silicon, B cobalt and C strontium tip. So this can really be an interesting study of angular momentum mapping. So students, let us summarize what we have learned in this module. AFM is used to characterize the morphology of the top surface of different kind of samples, including hard, soft, biological surfaces. It can operate even under liquids. AFM performs raster scan on samples in contact and non-contact modes, and vertical resolution is very good. Scanning tunneling microscopy involves a scanning method, but it requires conducting samples since tunneling current is used to map the surface. The lateral resolution of STM is very good. Even single atomic positions can be mapped. STM rather maps the electron density of the surface. Therefore, angular momenta and orbitals can also be mapped using STM. So students, we have just here you see that in this module discussed the 2 basic kinds of scanning techniques, but there is much more to learn, and to learn that we have to go to the references provided in the material, and it's extremely vast subject, interesting subject in the surface physics area.